So I'm Amy Smith, and I'm actually a postdoc at Hui right now with Julie Huber. But what I'm going to present to you today was part of my dissertation defense in November. And I am now part of the Subsea project, and we'll be going to Loihi Seamount soon, and I'm very excited about getting more involved in astrobiology in the future. So over here, what you see in the background are some microbes. I don't know if you can make them out, but I colored them so you can see them a little bit better. These microbes are attached to a surface of olivine that was incubated in the subseafloor on the Juan de Fuca ridge. So that temperature of the fluid is about 64, 65 degrees Celsius, and these microbes are presumably from the fluids, and they were colonizing the surface of the olivine over a period of time. So I spend a lot of time with these microbes, actually probably more time with these microbes and thinking about what they're doing than I spend with my children. So over time, they kind of start to talk to me, and I feel like they have some secrets to tell, and these microbes definitely have some secrets to tell, and they taught me a few things about life. So you can see that, that from this image, there's at least a few different types of morphologies of these cells. And generally, as microbiologists, that doesn't really tell us anything about who these microbes are. We can't really tell if they actually are different from each other or not. So we have some techniques that we've developed to help us decide who belongs with who has what sort of pathways, and is able to do certain things on this mineral. So, thanks. So, whoops, wrong one. So we know that iron exists in the ocean crust, and there are lots of iron-rich minerals, including olivine, in the ocean crust. And as water is reacting with those minerals, you can produce hydrogen. And this is a very simplified way of looking at it. But what I'm most interested in is what happens to that hydrogen, and what are the microbes doing with it. So there's also a possibility that they're using this iron as well, or iron oxides that could be produced from this reaction. So initially, when I was looking at these microbes, I thought that they might be using that iron, or the iron 2 or the iron 3 in the mineral itself. But it turns out that they're not. They're actually using the hydrogen, which is interesting because this is not really a serpentinizing environment as you think of it. But perhaps this is occurring. So you can see this is my kind of diagram of what this cell might look like that can use this hydrogen. And there are a few different pathways that microbes can use hydrogen, and some are able to use to build biomass using this hydrogen. Some are able to get energy from it, and some can do both. So my main question is, are they able to use the iron, or are they able to use the hydrogen? What are they doing on this mineral? So how we got about that was to do a 4-year subseafloor incubation of this olivine mineral. So we took sand-sized grains, and we put them in these little flow cells and packed them in there with some little sponges. You can see the sponges here. And over time, you can see there's some precipitates that kind of formed on the sponges, so there was reaction with the mineral and the water. This was also attached to an osmopump, which was pulling fluid through the minerals for the entire time, so that we know that the microbes that came from the fluid were getting onto the minerals themselves. And then what we did with this flow cell is put it inside one of those corks. This was on the Juan de Fuca ridge again. This is hole 1301A that we used for this experiment. And basically what you do is you put these flow cells and you suspend them within this cork into the basalt basement. And then you pull the fluid through for a period of time, and then you go and retrieve it. And I looked at the microbes that were attached to that mineral over that period of time. So this is a map of the hole location, 1301A. This is the water depth here. And the depth which we incubated these minerals was about 262 meters down into the basalt basement. There's a lot of sediment here, so you have to drill down pretty deep to get down into the sediment, or sorry, down into the basement. So the temperature, again, is about 64 degrees Celsius, so it's kind of a low temperature or moderate temperature. There is a little bit of hydrogen already in the fluid, so it's about 2 micromolar, but that is enough for hydrogenotrophy, but when you add the olivine in there in the equation, you might get some more. So I'm really curious as to how that might change the microbial metabolism. So some other substrates that were in the fluids as well, we had sulfate, ammonium, iron 2, and some nitrate as well. So those are all potentially used by the microbes. So what we do is take these microbes that are on the surface of the mineral and we shake them up really good, get them off the surface of the mineral, and then we extract their DNA. And then you have to break them all apart, all their genomes, you have to break them all apart into these little bitty tiny pieces so you can sequence it. And then what you have to do is amplify it and assemble it back together. And so what you end up with are these different length contigs, is what we call it, continuous sequences of DNA. And then you have to put those all back together and figure out which one goes to which genome, and then kind of look at their metabolism from there. So this is what we do from that point. We take those assembled metagenomes. A metagenome is all of the genomic material within that community. And so you take it all and you put it back into these genome bins. And so this is all of the different DNA you get from the different types of microbes, presumably. And then there's a lot of magic and a lot of math that happens here, a lot of computing. And then you can look at what's in these genome bins and figure out what enzymes can they make and what pathways do they have that are complete. So this is what your map of the genome bins kind of looks like in space. So you can see here for this olivine metagenome, we have 12 different types of genomes that we obtained. And so we've got 10, 11, and 12 here are archaea, and then the rest of these are all bacteria. And I just want to note that this one genome here is not actually a genome. It's part of a contaminant from the DNA extraction kit that we use. And that's been shown multiple times, and it showed up in several of my data sets. And then I'll show you it doesn't actually have pathways either. So it's just kind of junk that we have to deal with because all of these samples are really low biomass, and you can get contamination from the lab that will, mess up your sample. So I just left it in here so you could see the results of that as well. So I just want to point out here that, whoops, I knew I was going to hit the wrong one. So we've got those 3 archaea, and these are all bacteria. This number 4 and 5 are just bacteria. That's all we know about them. We know that they're bacteria and not archaea, and we don't know anything else about them. Never seen them before. The rest of these are Clostridia, which are deep branching bacteria. They're anaerobes, and some of them can survive high temperatures, as well as these archaea that we got are also hyperthermophiles and thermophiles. So that kind of matches with what we should expect from this aquifer. So this is what the results of the carbon fixation and energy metabolisms are from this community. So first, I want to direct you to this up here. So these are the bin numbers. So you can see that 10, 11, and 12 are the archaea, and the rest of these are bacteria. This number 1, if you look down, there's no complete pathways here. The complete pathways are the darker circles of each of these. And then I've broken these into nitrogen metabolism, methane and related pathways, so not necessarily methanogenesis, but pathways that are related to it, sulfur metabolism, and those other carbon fixation pathways and those that are related to it. And then down here, you can see the percent completeness of each of these genomes. It's pretty high. Most of these are 90 to 100% complete, which is amazing, really amazing. But I think it's because we only have like 12 genomes. Usually you have a lot more than that. So it's not a very diverse community. So let's look first at the nitrogen. We have one organism that has a complete pathway, and it's a nitrogen fixation pathway, which is pretty cool because a lot of times in a microbial community, you have to have a nitrogen fixer to be able to get organic nitrogen to the community. They have no other way of getting organic nitrogen. And then if we look at this methane-related pathways, we see that we don't have a complete pathway for methanogenesis, either from CO2, from acetate, or from methanol, no complete pathways. There is a complete acetyl-CoA pathway, which is part of the methanogenesis pathway, but these are in the archaea, and that's to be expected. For these archaea, they usually have that, and I'll show you an instance of that in just a second. But this pathway is actually a pathway where you can fix carbon as well. We have 4 genomes that have the assimilatory sulfate reduction pathway, so they can use sulfate reduction to gain energy. They can probably use hydrogen as well, or they can also use organics to do this. So that's a good energy pathway that's represented within this community. And then now if I direct your attention to the carbon fixation pathways, you see that this Wood-Ljungdahl pathway, like what is that, came up. And I didn't even know necessarily what this pathway was before I came into this, but now I know all about it, so I'll tell you about it. And then this here is the Calvin cycle, which is normally found abundant in crustal rocks at the surface. But this is also an indication that you have oxygen present, and the Wood-Ljungdahl pathway can only operate in anoxic conditions. It does not work with oxygen. So that could be one of the reasons why we see it here. And then there are this one other little accessory pathway that allows you to make energy from this Wood-Ljungdahl pathway instead of just biomass. And so there's one organism here that has both the biomass producing pathway and the energy producing pathway. So that's bin 6 here. So I'm really interested in this organism, and I'll show you a little bit about it if I have time in a minute. Okay, so acetogenesis and methanogenesis are basically the same pathway with different cofactors that they use. And so I just want to point out that this is still the Wood-Ljungdahl pathway. Methanogenesis uses the Wood-Ljungdahl pathway, and acetogenesis uses the Wood-Ljungdahl pathway. But what I'm seeing here is acetogenesis and not methanogenesis. So there is a difference. And that acetyl-CoA pathway that I was telling you about that the archaea have, that's just this branch here. So these organisms have this branch or both of these branches here, but not methanogenesis. And then I just want to point out one other thing here is there's this, this gene, this cofactor here called ECH that is active in allowing these organisms to get even more energy. In some acetogens, they use RNF, and some they use ECH. And so I'm seeing ECH in some of these organisms. So just to summarize the Wood-Ljungdahl pathway, most of these organisms that are performing acetogenesis are Clostridia, which is what I saw. They're very deep branching organisms. When I say that, I mean this LUCA should be over here. The last universal common ancestor between all of bacteria, which is represented here, all of archaea, which is represented here, and eukaryotes, which we occupy. That last universal common ancestor there, that's what it's really close to on the tree of life. So this pathway has a lot of important features that might be relevant to the origin of life or to life on other planets. So it's a really ancient pathway that arose early on Earth. It's bifunctional, so they can gain carbon and energy from this pathway. It's very simple, uses hydrogen and CO2. We see it in deep branching bacteria, so probably early life on Earth had it. And then recently it's also been found in some serpentinizing systems and deep subsurface environments. So I'm not the only one that's seeing this. This is the first time we're really seeing it in oceanic crust. But I think part of that is because it's attached to a mineral and not in the fluid, because most of these organisms that I'm seeing were not in the fluid. They're only on the mineral. Okay, so this is that ECH gene I was telling you about here. What you're seeing here are the copies of hydrogenases. So these are the enzymes that can break apart hydrogen, and you can use them for energy. And so the different types of hydrogenases are color-coded here. So these are all the subunits that code for this one enzyme. And then all the pink ones here are another one. So the blue ones here are ECH. And so we have at least 2 organisms, especially that bin 6, which has that hydrogenase, which means there is a really good likelihood that it is an acetogen. So if we look more closely at this organism, I'm not going to get into this really complex pathway and how it all feeds together, but I just want you to know that acetogens, because they don't typically get a lot of energy from acetogenesis, They particularly might have other pathways that they can use, like sulfate reduction, or maybe they can use glucose or something like that. But this organism, this bin6, doesn't have any of those. It only has the Wood-Ljungdahl pathway, and then it also has a couple of importers for small peptides and small amino acids, and that's it. So what I think is this organism might be an autotroph, and then it's feeding the rest of the community. That's at 10 or 15? Okay. Let me see. So this is just my idea of what's happening in the community. So I think this bin6, this organism, is using hydrogen and CO2 or bicarbonate in the seawater to produce acetate and help sustain this whole community. And then there are other players as well, reducing sulfate and using fixing nitrogen as well. And so the next steps is we really need to get these organisms in culture. I tried culturing microbes before, but I wasn't successful because I wasn't expecting this pathway. And so now we know we have this information, so we can go back, maybe, to the Juan de Fuca and try to get these organisms in culture and see if they're actually doing this or not, which is really important. So we have some more questions, like what are their energetic limitations? And are there some biosignatures that these organisms may produce that we can look for on other ocean worlds? Okay, thanks to lots of people. And I'll open it up for questions now. Thanks for listening.
Amy, nice talk, especially being put on the spot to go first when you thought you were going second. Two relatively quick things. First thing is, how do you know that the microbes that you were examining here were the ones that were alive and metabolically active? Did you look at any ATP, or is there a way to differentiate that? And then second, on the initial image, it looked like all of your microbes were loners, out on their own, doing their own thing. Is that pretty typical of what you saw, or have you seen consortia of the bacteria and archaea working together?
I don't necessarily see like clumps of bacteria. There were some, I incubated a whole bunch of different minerals, not just olivine. And in some cases you see big clumps, like they were dividing all in the same area or something like that. But yeah, you don't necessarily see them actually dividing or necessarily working together. But they do have this big biofilm that they're in. I did some other work to show that there's a carbon-rich biofilm on the surface. And so there might be something going on there to help the microbes. Yeah. No ATP measurements.
Great talk, Amy. I don't
Thanks.
think I've actually seen you present this, so this is great. I'm going back to Juan de Fuca next year, so let's talk about
Okay.
culturing. That would be great.
I think it's important.
Yeah.
Awesome talk. So if you could sort of do this experiment instead of just, incubating for a long time and going back and collecting it all at once, but instead as like a time series, who do you think would be colonizing first, and then maybe creating things that other things could move in and use?
That's a really great question. So because a lot of these organisms aren't necessarily abundant in the fluid, I would imagine that they would not necessarily be the first ones to colonize. But I don't really know. There are a lot of spores in the fluid as well, so it's possible that a spore would sit on and then start, growing, germinating. Yeah, I don't really know. And actually, I can tell you when we first drilled this hole and put the minerals down, it didn't completely seal, so there was seawater mixing for a while, for like 3 years. So there was potentially some other things happening as well before these microbes, kind of got sustained in their community.
is there a way in the genomic data to distinguish whether that may be what's going on?
Yeah, there's a lot of questions that I'm still trying to work out a lot of it. But this one particular organism that I'm looking at, it doesn't have any importers for any lipids or carbohydrates at all, and only those small peptides and amino acids, certain amino acids, the only 3 amino acids. So that makes me think that there's something else going on there. There's this thing called the Sticklin reaction, where they can use 2 amino acids to gain energy, like switching electrons. They're using one to reduce the other, and so they could potentially be using that as well. But I couldn't find the enzymes in this organism that are typically found. But that doesn't mean that they're not there, or that we don't really know.